So without further ado, I can introduce our first speaker, Dr. Shehla Mohammed. So Dr. Shehla Mohammed has been a genetics consultant for the BBS clinics held in St. Thomas's Hospital, London, since their inception 14 years ago. Shehla is a passionate advocate and ally of the rare disease and Bardet-Biedl syndrome community and has served on the BBS UK Board of Trustees since 2021. Shehla is going to give us an introductory overview of Bardet-Biedl syndrome. So Shehla, thank you for presenting for us this morning. Over to you.
Lovely. Thank you so much. Good morning, everyone. It's a real pleasure to be here and really delighted that so many friends, colleagues, patients, advocates could join us. I was asked really to just give you a little bit of an introduction and background to BBS. We have a really exciting program to update you on all that has been achieved in terms of novel work, understanding new therapies on the horizon. And I appreciate although this is a small talk, you know, many of the principles will be familiar to you, but just this is just a bit of a refresher to BBS. So on this slide is just really an outline of what I hope to talk in the next 10 to 15 minutes. So we have a young girl looking at a DNA helix on the screen, and what I hope we can cover is just some of the basic concepts around genetics, inheritance of BBS, and a little bit of reflection on the many ways that a patient with BBS can present to us in clinic. And also ready to go through some of the commonly asked questions that we have in clinic, the multidisciplinary clinics that we run in the UK. So, as you all know, we have, this is a bit of basic biology. In each cell, we have a central area called the nucleus, which has our chromosomes, which carry our genetic information. And within the chromosomes, we have the DNA coil really tightly coiled upon itself. And this carries all the set of instructions that tell the body how to grow and develop. And it's important that the set of instructions is complete. We all carry some minor changes in our genetic makeup, but we're just going to focus on BBS. Many minor changes don't make any difference, but for BBS, we know that there are specific genetic changes that can have an effect on the function of many systems. So, what is BBS? We recognize this is a very rare genetic condition. Many people may not have come across the condition, but increasingly with the level of awareness that has been possible over the last few years, perhaps it's not such an uncommon situation, but nevertheless it remains a rare condition which has an impact on a number of different systems, predominantly on sight loss and a number of other systems. It's fair to say that the condition can vary enormously, and on our present information that's available, we feel there are probably around 640 people who are known to have BBS in the UK. So the symptoms, as I said, can vary enormously, and some of them are listed here. So people can experience sight loss. They have a tendency often to gain weight because they feel hungry. Some patients may have learning disabilities, and they may have other physical features such as kidney problems. They may have hormone issues. Some patients may struggle with speech and learning. So we recognize that because there is so much variation, sometimes a clinical diagnosis can be quite difficult because individually these symptoms can occur in people even without BBS. But many years ago, it was possible from our learning and primarily from the work of Professor Beals and other colleagues to be able to sort of group these symptoms together, which could enable us to make an easier diagnosis. And these are listed on this slide. So there are primary or major features or secondary features, which reflect the involvement of the eyes with the rod cone dystrophy, extra digits, learning disabilities, proportion of renal abnormalities, and as well as some secondary features along with an effect on balance, learning, and diabetes. So Professor Beals, back in the day, I think it was about 1909, when he was doing his research, he was probably about 18. He certainly looked 18 at the time. Described these features back in 1909 about the combination of problems that we see in individuals with BBS, and then subsequently in 2013. Elizabeth Forsyth and Phil Beals looked into the proportion of problems that could be seen, and they're listed in this sort of long list that I've mentioned. But essentially, the combination of 4 of these major features, which are at the top of the slide, or 1 minor or 3 major and 2 minor features, help to make certainly a clinical diagnosis. So the mode of inheritance is well recognized as being autosomal recessive. So it's inherited in a recessive manner, which basically means that we all have 2 copies of each gene. So we inherit 1 gene, 1 pair of genes from our mother and 1 pair of genes from our father. So in this cartoon, we have a carrier parent. So this is represented as, on the blue side is the normal copy. The orange side in each parent is the one that's got the spelling mistake in a gene. And in recessive conditions, both copies of the gene have a change or a spelling mistake, or what's called a mutation in a person, in order for them to develop the condition. So when a child has BBS, we would expect both parents to be carriers. So they have 1 normal copy, and which is the healthy copy of the gene, and 1 with the spelling mistake. And being a healthy carrier has no implications for that person's own health. However, if 2 carrier parents come to have children, at each pregnancy, there is a 1 in 4 chance, or a 25% chance they would have a baby with BBS, and a 75% chance that the baby would not have BBS. So they would either be not a carrier, or they could just be healthy carriers. So as you will all know, over the years, many BBS genes have been discovered. At last count, I think there were about 22 genes that we can now test for, so we can look at the precise spelling mistake in a gene. So in this picture, you have what you see is a circle with different colored segments, and these different colored segments represent different genes from BBS1 and upwards. The most common genetic alteration that we see is the one represented here in blue, and that is the BBS1 gene. And then the next more common one is the BBS10 gene. So essentially in clinic, if we see a patient with BBS, And the clinical features that I mentioned earlier seem to suggest that is the most likely diagnosis. There is an 85% chance that we would be able to identify the precise cause of BBS in that person. About 40% of cases, BBS1 gene has a spelling mistake, so that's the most common one that we see, and the next common one is the BBS10 gene. So what that is indicating is that there are a proportion of patients where we still don't know what the underlying genetic cause is. And I'm sure over time, these will be found due to work that is ongoing by our clinical scientists. So you may be aware that BBS is described as a ciliopathy. And I won't go into a lot of detail because it may well be covered in some of the subsequent talks. But BBS1 is a ciliopathy, and one of the questions we often get asked is, well, what are cilia and why are they important? So cilia are these long, very thin hair-like projections on the surface of the cell, and there are two different types. The ones that are important are the ones that affect sort of sensory cilia, and they are present in many organs in the body. And with regards to BBS, the most common one are the cilia that are present in the eye, in the retina, the back of the eye, which are the photoreceptors, and also in the kidneys. And sorry, I don't know why this is jumping, but anyway, so we recognize that the spelling mistakes in BBS gene can affect different people in many different ways. So when you read the information, and there's a lot of detail in the medical information booklet, it lists everything that has ever been noted, described in patients with BBS, but that does not mean that all of those aspects are relevant to individuals. So we can see patients with, say, the more common BBS1 mutation, and some of them will have an extra digit or polydactyly, whereas another person with exactly the same BBS1 mutation may not. So, and similarly for all of the symptoms, there can be quite a lot of variation in presentation. So we, in clinic, one of the more frequently asked questions is really about the wider family. Understandably, parents may wish to know whether some of the other children that they have, what the implications may be for them. So if we know, as seen in this, so you've got a diagram here with some circles and a little red spot in the middle, and that represents a person who's a carrier. And a full circle here is an affected child with BBS. If we know the precise genetic alteration in the BBS gene, then it is possible for us to do a carrier test for siblings if they wish to have that information. Another question that will sometimes come up in clinic is if a young couple were planning a pregnancy in the future, and they will inquire about what options may be available in the future. Now, planning a pregnancy where there may be the option of testing is possible, is a very, very personal choice, and whatever choice couples may wish to make, we are there in the clinic to support and explain what the choices are and support their decision. So one option may be not to do any testing whatsoever, and that's completely understandable. Some couples may say, well, we don't want to do anything that may be an invasive test in a pregnancy. Could we just have a detailed scan to look for some of the features that can be associated with BBS? So that is possible, but there are limitations of that testing, and it's important that any kind of detailed or specialist scans are undertaken in a unit that has specific knowledge and expertise. So extra digits or fingers or toes may be picked up, but other features may be very, very subtle, particularly if they involve the kidney. So there are limitations to scanning, and it may not be possible to pick up those aspects just on even on a detailed scan. There is also the option of actually testing the pregnancy, either taking a small sample of the placenta, which is called the CVS test, normally done at about 11 weeks, or an amniocentesis test done at 16 weeks, where we take a tiny bit of fluid from around the baby, and then the laboratory then analyzes the information to see if the genetic information known to be present in the family can be picked up. Another option may be to do pre-implantation testing, or what's called PGT, and this is this is a process whereby couples can undergo an IVF type of procedure, but where the genetic testing is done in the laboratory, and only those embryos are implanted back in to the mother that are known to be unaffected with BBS, but it is a complicated process. Some couples may opt to just test the baby after birth, just using a sample taken from the cord after delivery. So there are a number of different options, as I've mentioned. So we have learned a huge amount over the last few years. When the initial clinics were sort of set up back in 2010, and these were set up with the pivotal support of BBS UK, commissioned by NHS England in London and in Birmingham. And it's fair to say we feel that they have made a really huge impact in enabling access to patients with BBS, making sure they have the relevant support, information, that we can coordinate their service. And we have learned a lot from being able to see patients over a period of time. And many of the talks that you will hear today is really a reflection of being able to see patients with BBS and learn more about the condition, which have enabled these new understanding and new therapies to come online. So I'm happy to leave that at, on that last slide. And I think, as Tonya said, we'll do talks, answer any questions perhaps at the end of the session. Thank you very much. Thank you.
Thank you so much, Shehla. That was hugely informative and a great introduction to the day. So Shehla said we'll be having a Q&A panel at the end of this first session.